This is Mr. Wartsky again, and this is the second screencast for the immune system. This is the last slide that you took a look at before. Take a look at it again if you like it. Great, take a picture. So the second or the third slide for the third line of defense, which is the specific internal, T and B cells are both lymphocytes. So when they are stimulated, they will undergo clonal selection, which means that they will undergo rapid cell division and produce effector cells. In B cells, the effector cells are known as plasma cells and B memory cells. Plasma cells will produce about 2,000 antibodies per second for about 4 to 5 days and die of exhaustion, whereas the B memory cells will just wait and wait and wait, and they'll wait for a second time that you're exposed to the same antigen, and then they will be able to produce plasma cells almost immediately. This will cut down your response time from 10 to 17 days to 2 to 7 days so that you can wipe out the pathogen before you get sick. Because remember, you get sick or you feel sick because you have a histamine response because the pathogen is killing the cells in that area. Now, the T cells also have two effector cells. They produce TH, are called T helper cells, and TC cells, which are called cytotoxic T cells. T helper cells will help the B cells produce antibodies, where the TC cells will kill infected cells by poking holes in them. Now remember, T and B cells originate from the bone marrow. B cells will mature in the bone marrow, and T cells will mature in the thymus with help from thymosin. That's the hormone that the thymus produces. Now, how does our body recognize invaders? Now, on our cells, we have these things called, these proteins or glycoproteins called major histocompatibility complexes, or MHCs. Each MHC is coded for by 20 genes, and each gene has 50 different possible alleles. So this protein or glycoprotein tells your body that the cell belongs to you. The cell belongs to you. So there are two types of MHCs. We have class 1, which are on all nucleated cells. Now, interestingly enough, the red blood cell has no nucleus, so it has no MHC. The class 2 MHCs are on B cells and macrophages. The function of the MHC is called antigen presenting. The cell will take in the antigen, break it apart, and put part of the antigen in the MHC and present it to the rest of the other cells. Okay? And so it will hold the antigen and present it to T cells, TH cells, as well as TC cells. So here the class 1 MHC, class 2 MHC, the antigen will be here, and now those are antigen presenting cells, or APCs, antigen presenting cells. So here we go. The antigen gets past your external defenses. The antigen gets past your nonspecific internal defenses. It will float in your blood and it'll eventually be forced into your lymph system, which is a secondary circulatory system that runs parallel to your blood vessels. The lymph system, the fluid will flow to your lymph node because of muscle contractions. Once in the lymph node, the antigen will be exposed to high concentrations of T cells, B cells, and macrophages. And it happens in your lymph nodes. So in the T cell dependent production of antibodies, which happens most frequently, the B cells take in the antigen. And you need to slow down on this because there's a lot of things happen. Pieces of the antigen will be incorporated into the class 2 MHC, and it will be an antigen presenting cell B, a B cell that's antigen presenting. I say APC-B. Usually, the B cell won't go through clonal selection if the antigen is too small, and that's what happens most of the time. T cells with the correct receptor will bind to the antigen right? The T cells will undergo clonal selection and they will produce two effector cells, T helper cells and cytotoxic T cells, TH and TC cells. Now we're going to focus on the TH cells only for now. We'll get into the TC cells in a moment. At the same time, the macrophage takes in the antigen. Pieces of the antigen will be present in the class 2 MHC. It is now an APC macrophage. In addition, the macrophage will produce a cytokine called interleukin-1, and that can act as a pyrogen, which can cause you to have a fever, reset the hypothalamus, and can also stimulate the production of IL-2 in T cells. So that's a lot right there. Make sure you get it all down. If you have questions, you can rewind this and listen to me talk again. So far, this is what we have, right? Macrophage is taking it in, presenting. The B cells taking it in, presenting. The T cells, oh, where are the T cells? Have produced T8 cells. T8 cells. Right? Beautiful, beautiful. A T8 cell with the correct receptor will recognize and bind to the APC macrophage. Now, there's a protein on the T helper cell which is called CD4 and helps with the binding of the TH to APC cells, or antigen presenting cells. The TH, when it binds to the APC macrophage, will undergo clonal selection to produce more TH cells. Now, the interleukin-2 also stimulates the production of TH cells. So we basically have an explosion of TH cells, and it's a positive feedback loop. The stimulus creates the response that increases the stimulus. Because the TH cells produce the IL-2, which causes them to divide rapidly. So they produce more IL-2, so it causes them to divide more rapidly. They produce more IL-2, which causes them to divide more rapidly. That's good. Now, TH cells will bind to the APC B cells. When bound, the B cells will undergo clonal selection and produce plasma cells and B memory cells. Now, IL-2 will also stimulate B and TC cells to reproduce too. It's IL-2, man, it's a mixer. Now, plasma cells will produce 2,000 antibodies per second for about 4 to 5 days. And the more plasma cells you have, the more antibodies you produce. Now, the antibodies will end up in your blood and start binding to every antigen that they can. Now, in T cell independent, you need a very large antigen to bind to B cells. And when that happens, you can cause them to produce plasma cells, but the response isn't as strong as T cell dependent. So we're going to focus on T cell dependent whenever I ask you to talk about the production of antibodies. So how do antibodies work? Well, first it's called neutralization. So the antibodies will bind to the viral binding sites, and if the viral binding sites are all bound up by antibodies, they can't get into cells. Then what happens? The macrophage will eat the antibody viral complex, of antibody virus complex. Agglutination, in which you have an antibody binding to an antigen, which acts as a flag, and a macrophage will come along and eat it. Then you have precipitation, in which many antibodies bind to many antigens, and then the macrophage comes along and eats it. Some antibodies can carry hydrogen peroxide with their constant region, and then that can destroy the bacteria. Now, there is a technique which is called monoclonal antibodies, where you combine a plasma cell with a cancer cell, and then you can produce the antibody indefinitely. So here's how antibodies work. They bind to the virus, neutralization, agglutination. They bind to, and then the macrophage eats it. Or precipitation, a whole bunch bind to it. They form this chain, then the macrophage eats it. Now, in the cell-mediated response, So this happens when the cell is already infected. So cells with the antigen, just a normal body cell, will express the antigen in the class 1 MHC and is called an APC cell. Okay? Antigen presenting cell. The TC cell with the correct receptor will bind to the APC cell, and a special protein called CD8 will help with the binding of the cell to the TC cell. The TC cell will produce perforin, and a hole is poked in the cell membrane. The cell dies, the antigens are leaked out, and the antibodies bind to it. That's a lot shorter. Cell-mediated response. Cell binds here, pokes a hole, leaks out. Now, how do we apply this stuff? Well, blood groups A, B, and O, the red blood cells do not have a nucleus, so the only protein that we have to worry about matching is the A protein, the B protein, or the lack of protein. Okay? Since there are no MHCs, we don't have to worry about tissue typing, because that's what we do when we tissue type. We look at the MHCs. So that's the next one, tissue transplant. The MHCs, we're going to cause the body to respond to it with the immune system. So you need to suppress the immune system for the entire life of the individual. And that can be done with cyclosporin, which is produced by fungus to suppress the immune system. Autoimmune, rheumatoid arthritis, insulin-dependent diabetes, rheumatic fever, in which you attack your heart valves or heart muscle. Graves, you attack the thyroid. Lupus, you attack everything. All body systems are attacked by lupus. Multiple sclerosis, the myelin sheath on the nerves, and alopecia, skin cells, vitiligo. No, alopecia is hair cells. Vitiligo is melanin pigment cells. So what happens is your body attacks those cells and kills those cells. Don't know why yet. So we have the ability to produce antibodies and immune response for every cell in our body, but something shuts off those T cells and B cells before they mature. Sometimes that doesn't work. Allergies. In allergies, what happens is the mast cell will bind to the antigen and release histamine. Now, in anaphylactic shock, a lot of histamine can be released because the antigen gets into your blood, travels throughout your body, binds to the mast cells. They release their histamine. All your blood vessels dilate. When all your blood vessels dilate, your blood pressure decreases, your heart beats rapidly. Eventually, you will die in approximately 15 minutes. And so that's bad. Now, immunodeficiencies, HIV/AIDS, you know that they kill T helper cells by binding to the CD4 protein. Yep. And they think that the cytokines are chemicals that actually make you act like you're sick. That type of depression to decrease the energy usage so you have more energy to fight the disease. Now, if you have any questions, by all means, read your textbook, watch the video over again, or ask me in class tomorrow. Have a good night.